what I want to do here, what I always like to do is to share the pain of dealing with taxonomy, because probably like many of you, I spend a lot of time with a microscope staring at bugs and looking at keys and being deeply puzzled until sometimes I find the answer. I want to sort of outline what I see as the issues with using keys for non-specialists, which I certainly count myself as. And then I'll show you a couple of examples of the key building software that I've played with that I think get at some of those issues. And then I'd be delighted for feedback on this. So we can talk about that more a little later. Next.
And I'm just going to interrupt for 2 seconds to say,
Sure.
you know, I'm often managing both the chat and getting Q&A from the emails. And just to let y'all know, while I'm doing this screen share, I can't look at my email. So send questions later. But chat, you're good to go.
Okay, good. Okay, so here's why I'm involved in this game at all. Yeah, my background is in everything other than bees and pollinators. I worked for a long time on emerald ash borer and various other out east pest issues. But now I've spent the last 3 field seasons studying pollinators in the Willamette Valley, that's in Western Oregon. And there's 2 projects that I work on. One is the evolution of pollinator communities in prairies after they've been restored. So what happens to pollinator networks? And the other one is understanding the breeding system of an endangered species called the Willamette daisy. And so we're learning everything we can about its pollinators. Next. So the network study, I mean, I'm not going to get into the weeds, so to speak, on these things, but just to give due credit, Susan Waters is the investigator for this network study we're doing. And her mission is to create these wild spaghetti charts. And what she's trying to do is figure out what happens at the ecological sort of system level to the whole suite of pollinators and the plants that they visit. And many of you have seen these diagrams, I suspect. And she's looking, actually, I'll just say briefly that each one of these little pictures here shows you what happens with pollinators across the top of each little graph, plants across the bottom, and arrows that connect them proportional to the visit intensity of that particular pollinators. And you can even see in this little tiny set of charts that over time, after a prairie has been restored, it gets way more complicated and many more relationships. So that's the one thing that we work on. And we collect pollinators on every plant in about 14 different sites in Washington and Oregon. So we have a lot of data from that. Next slide. Claire, are you moving to the next slide? There we go. Well, okay. The other project that I've been playing with is to look at the pollinators of Willamette daisy. And in this one, we go out and we watch the tiny patches of this endangered plant. I've probably seen, 80% of the plants in existence. And we run around and we collect everything that visits these flowers. And there's some other aspects of the breeding system of the plant from a botany point of view that we do. So next slide. So like in any project of this sort, we end up with 100 boxes of bugs. And the challenges then are to try to put a name on everything. It's only useful if we can, figure out what the real diversity is. And if we can't put a name on it, which is disturbingly often, we will call it a morphospecies whatever, and we need to sort of keep track of that, because to some extent the species richness can be understood if we can discriminate morphospecies. If we don't have a name for it, we can still say that there's Andrena is really diverse and Megachile not so much, that sort of thing. And then another challenge for me is how can I help my colleagues, sometimes, not people who've spent a lot of time with keys and thinking about bugs, how can I help them do a rough sort of the specimens we collect, at least to genus? So the options for dealing with all of these challenges are I can go to experts, and my best intelligence says that if I try to send Sam Droege or Lincoln Best out here in Oregon, or you name it, expert, they're going to be jammed up because everybody wants names for their bugs. And so maybe I'll send stuff off and it'll sit on the edge of someone's lab bench until they get to it in 2024. At least I'm warned that that is one outcome. Another thing you can do is you can barcode specimens, and we do that on a limited basis, but at some point it becomes too expensive to do every mystery. So you're left with learning to navigate diagnostic keys, and I'm a very good test case, a motivated non-specialist without a big reference collection. What can I do to figure out these taxa from the keys that I can easily access? Next slide. So I'm just going to say a couple of the issues that I have with keys. An important caveat here: I'm not critical of keys in general. There's way too many things to figure out, and limited material and limited time, and so there's just a huge suite of different solutions to this problem of diagnosis. And they're all really valuable. And I'm highlighting Discover Life here, partly because I know that some people have skin in that particular game here. And I think it's a really good and helpful key. And it's my go-to for a number of things. But it does have a couple limitations for me. One is that, as you see in a little picture down here below, every time I start to work through a key, I try to find Oregon in the checkboxes for where I collected my B, and disturbingly often there's no place for Oregon. And then the converse is that keys are often over-inclusive from my perspective in Oregon. So I'm pretty sure that we have 8 Ceratina in the Pacific Northwest, and there's 46 in the Discover Life key. So I'm going to have to weed through a lot of things that are just not going to happen for me. So it makes it a much more daunting process for me to get those. So next slide, more issues. So here's a really big one, and I'm sure all of you have run into this, the crisis when you get to a couplet in a dichotomous key, and you have to know which way to go, and if you go the wrong way, you'll never get the right answer. And my go-to example for this is in the McGinley key to Lasioglossum, which is, from a technical standpoint, I'm sure it's the go-to key as far as I know, and it's really, really good. And if you start working through it, at the very beginning of the key, you're asked to make some choices about the Acarinarium. Well, this is a hard feature to figure out if you're a novice. And the picture that you see here, it's not super helpful because I don't have the detail to see there. And a further problem is that for lots of the Lasioglossum that we have, they've been mounted so that you do not see the acronym. It's if the abdomen is not somewhat depressed, you can't look at it. So I'm stymied right at the get-go with this key. And there's going to be further stumbling blocks if I get further in this key. And again, this one is for Lasioglossum north of the Mexican border. So I'm going to be looking at a lot of species that we don't have, and I shouldn't have to think about. So next slide. And then a final issue with keys for me is the use of both diagrams or photos of features and of the destination taxa. So there's a huge range in the usability of these things. The worst case would be like a little tiny pinned insect that has no features that I can make any sense out of. And then there are sometimes better images of whole insects, but they're kind of crumpled up and they were just taken out of a drawer and bang, they were shot. And, that's somewhat helpful. Better, I like the images that, maybe are higher quality, and I like the feature where you can zoom in and maybe look at some piece of the anatomy that's going to be helpful. But what I really want is the next slide. So this would be super helpful if you were building a key, and also if you were trying to figure out a key. If you could see what it really looks like through a microscope, and if you could, look at the pits and count them, if you could maybe look at the wing and, measure the different pieces of it and so on. So in a perfect world, keys would, when you get to a destination, is it taxon X? You'd be able to look at a picture like this, and this gets you a little bit past just the details and says Gestalt. Is this look kind of like what I've got? Despite whatever the character sets looked like as I went through the key. Okay, next slide. So thinking about all this stuff and knowing about lucid keys, which are interactive, there's a really nice one on exotic bee ID. There's a few of these things out there. And I thought, I want to be able to make my own key for particular purposes. So I hunted around and I found a couple models for creating my own key. And I want to be very clear about what the limitations are here. I'm not personally going to try to do a dialectus of the world key. There's, it's way beyond what I could do. So I want to be modest and say that my niche, the things that I want to do with a key are, one, tracking the taxa that we have within our own project so that I can communicate with another person in the project in another state what I mean by whatever taxon I have tried to name. And then I thought it would be reasonable for me to do the genus in a limited region like the Willamette Valley, which is where I live and where we do our work. So there's roughly 23 genera in the Willamette Valley. There's 45 in Oregon, and I don't have the material to even attempt that, but I think I can probably do a pretty good job here, and I can get the people that I work with to genus hopefully almost every time. And then the other niche for me is to take a relatively low diversity taxon. The one I'm working with right now is Ceratina, which, as I mentioned, has just 8 species. So that key should not be too hard to do. Next slide. Okay, so for project ID tracking, this is super useful for me. And I have an example using software from, I'm not sure what to call it exactly. They call it the biodiversity management platform, something like that. It's an organization based in France that makes these keys available, and they've been used for all sorts of taxa plants and such. And I use this to document the morphospecies that we identify in our project. So we have, I'd say, at least 30 Andrina, probably 40, which is super daunting. And I've done my best to, mainly using the Discover Life key, to walk through and say, okay, I think this is Nivalis, and here's why. And I have no idea what this is, but I know it has, pit diameters of X and Y, and it has fovea that are light and dark, et cetera, et cetera. And so once I go through the pain of figuring out how to sort the Andrina that I'm looking at, how do I tell my colleague in Washington what I mean by morphospecies 17? Well, if I can give her a key that lists all the characters that I use to make that designation, then whether we're right or not, we at least are sorting our bugs in the same way, so we can, go to the next level of ID with the same set of insects. And if I was on my own computer, I would browse around a little bit and show you these examples. I wouldn't really show you the Andrina key because it's slightly embarrassing. Megachile, yeah, I think I can tell a colleague in Washington what Megachile she has, because there's just not that many for us to look at. So that's this one type of key software. Next slide. And what I found in using this one, the advantages are that it's fairly easy to use, and it's entirely based on the web. So you do your key creation on your own desktop in a web interface, and then you upload it through some sort of magic through a server in France, and there's all kinds of JavaScript that's involved in piecing together a key and posting it online. So anybody could do this. We could figure out how to do it this afternoon. And interestingly, it also presents both a dichotomous and an interactive key. The dichotomous key, it uses some formula to figure out what's the most efficient way to use the characters that you've specified to get to some destination. And so that might be an easier way to go in some cases, but I tend to prefer interactive keys, which, as you know, having used Discover Life and some others, it allows you to sort of work on the characters that make sense to you, the things you can see. You don't have to look at that a carinarium. If you say, I can't see it, I can't enter that character, well, is the scape, longer than some other part of the bug, et cetera. So you just pick the characters that work for you. With this particular platform, I stopped using it because there's some things that just didn't work from a technical point of view. For example, if I had more than 14 characters I wanted to use, they would not display on the web page. So the user would not know that there were additional characters that they could choose. And in trying to get support for this, it was very sketchy. And it's based in France. And I kind of felt like, to be polite, I should probably send my emails in French, but failed at that, of course. So I went on to a different platform. So that's the next slide, I think. So I used a genus key, or used a different software called Identikit to create the genus key that I've mentioned. And this one is from an organization called the Field Studies Council in the United Kingdom. And the software has been used for all kinds of taxa, for harvestmen, for worms, and so on, and usually very nicely done. And so what I wanted to do with this key was get a citizen scientist or one of my colleagues to nail the genus for insects that were collected in the sort of habitats that we work in. So I'm not trying to say it's all across Oregon, because you get crazy diversity as you go up in elevation and so on. So we have a relatively simple set to consider. And I wanted to make a key like this without talking about subantennal sutures. That's sort of a sore point for me. It was a long time looking at these before I felt like I could even see them. So, for a lot of folks, I would prefer to find easier things to deal with. So next slide. So yeah, luckily I made a PowerPoint that doesn't require me to go on a browser to show you how this stuff works. So looking at the various red types here, starting from the left, for a set of characters, I can group them any way that I want, so I've tried to pick obvious subcategories here. And within each subcategory, to the right, I can look at the traits for that category, and then there's a little checkbox for what best fits. And if I hover over, for example, where it says Scopy for females only, if I hover over that, I will get a drop down, which you now see to the far left, which shows what I mean by that. So I can put help text in, and I can put images in to say, this is what I mean by brown or black. This is, if you're trying to do sex, here's a picture of the tergites, picture of the antennae, etc. And then on the right is one of the ways that I can view the outcomes of having chosen these various features. So the one I'm showing here is a single column key, and it shows to the right and the sort of tipped on its side legends there. For the characters that I've used, you'll see like length in millimeters. What's the score for that taxon for that feature? So if I said it's 10 millimeters, well, there's a lot of things that could be 10 millimeters. If I said the color of the integument is dark and non-metallic, you can see that a lot of things could fit this, a lot of genera, but a capostamen would not. So it gets at the bottom of this list a -1, so its score goes down, and so on. So I can just accumulate characters off to the right, and as I do, the weighted score becomes hopefully narrower and narrower, and I end up with the right answer. And then for any particular taxon, I can click on the picture, and I haven't really populated these, you know, to a great extent, but I can put whatever text I want into that descriptor, and as many pictures as I'd like. And what I do in my little info pieces for each of these taxa is I put a link to a URL that gets all of the Oregon and Washington iNaturalist observations for that taxon. So it's sort of a shortcut to say, if, you know, here's my pictures of Elasioglossum, but if you click here, you'll be able to see lots of other stuff that have been seen in our region. So give you a better sense of the diversity. So next slide. Another way I can display the information is just in a side-by-side comparison. So I pick on the left any taxa that I wish, and it will display for the characters that I've scored all the ways that they're similar or where they differ in the orange type there. So you can get a sense of how that stuff works. And I think this is useful if I have very few taxa that I really want to think hard about. It gets me down into it without being confused by all the stuff that the key throws up in the previous view. And then there's one other view option. Next slide. Which I really like. They call it the circle pack key, and this sorts things by taxonomic group. And so for this very simple example, on the left I have the different families that we have and the various genera within each family. And this is great for some sort of, for someone who's a novice to say, try to think about Megachilidae as a group because they have certain features in common, or Halictidae. And then if I pick one trait, metallic, you can see on the right that I have each one of those groups, and the big blue circles tell me that there are genera within Andrenidae. There's only 2, right? But there is a genus that scores really well for metallic, and likewise amongst Apidae, Ceratina is pretty metallic, and the other ones have little orange circles, which suggest they're not a very good fit, etc. So this is a fun one to play with. Next slide. So advantages that I found for this way of trying to create a key. Well, actually, a disadvantage is harder to learn than the previous one. But the documentation is excellent, and so it's figure outable. And there is a forum where you can get some information from the folks that created these keys about how they work or how to solve certain problems. And then it's an advantage and a disadvantage that the character states contribute to quantitative scores. So the upside of this is if I scored something incorrectly, or if the user scores it incorrectly because they didn't catch faintly metallic, they picked brown or black, but I thought it should be faintly metallic or whatever. As you accumulate more and more characters, the sum score, the average score, is going to converge, hopefully, on the right taxa, or 2 or 3 that you can make a choice about. And in every case, if you have really good pictures, you work through a key like this, and when you get close to the end, you start looking at the pictures and say, okay, the key says it should be Coleoxys. Does it really look like this picture? And then you know that you're pretty close, or that you're right on. Problem with this one is that to use this key at all, you can either put it on your own computer with software that you put onto your desktop, and then anybody who wants to use the key needs the same set of software on the desktop, and it's not easy. You have to create certain files and work in a command line interface and so on. So that's difficult. What you want to do is put it on the web, like I have, and that's not straightforward. You have to have access to a web server. In my case, I had to sort of jump through some hoops to use GitHub to create, a set of JavaScript files that delivers an HTML page. So it's a little complicated, and if you're lucky, you have a son-in-law who really knows how to do this. For myself, that would have been sort of a deal breaker if I didn't have that support. And similarly, there's some ways you can customize the key, the way it displays. You can choose, for example, which view, single column or whatever you want it to display, but you have to get in and edit files that it's a little bit scary because they're, scripts and they have interesting language. So next slide. So what I'm working on right now is the Seratina key. And it's an example of what I would call a bite-sized taxon. It's not super complicated. I think it can be determined without dissecting genitalia or getting to something that's going to be specialized and difficult. And the advantage of making a key for Seratina is, one, I know what the universe is, or at least I'm pretty darn sure that there's 8 species in the Pacific Northwest. So manageable group. I have a box with replicates of males and females for each one of those. This is pretty important. So, for my previous, like say the genus key, that depended on what I happen to have in our own collection. And I have a single Duphoria, for example. So I don't have a way to make images of or really think very hard about what the various Duphoria look like. A more egregious case would be Diadasia. So we have only one that we've collected, Nigrophons, I think it's called, and it has, and it's very black. And so if it was just me, I would say Diadasia is really black and hairy bee, but most of them aren't black, so I'd have to know that. So for Seratina, I will know that. And I also have, professional IDs on these Seratina. So it doesn't depend on whether I navigated a key correctly. Link Best, who's part of the Oregon Bee Atlas, is the one who ID'd these, and I have a lot of confidence in his ability to do that. And then finally, for Seratina, there's a fairly simple dichotomous key that I can use as a starting point. And my mission with that dichotomous key will be to figure out what parts of that are going to guide me in the right way and make me successful, and what parts are just details that won't matter for the 8 Seratina species I'm concerned with. So next slide. So my process for making a Seratina key is first I want a complete set of images. And usually the pictures I take of bees, I just do a lateral view. It's about opportunity cost. It takes a while to do these. So if I want to do a lot of taxa, I just do one shot. But if I want to be precise, I'll get the standard 3. So first thing is I'll get 3 views of each of the 8 Seratina for both sexes. Next slide. And then I'll look at the key that I have and get a feel for what the traits are that are discriminatory. So, a big one is what the hind, or this is, I think, a middle leg, what they look like, that little projection with hairs on it. That's going to be pretty important, I think, for males in particular. I haven't really studied this very much yet. I know that the punctation is an important clue. The picture up in the upper right is missionary, and it has very dense punctation. That's going to be a character I know I can use. On 2 of the 8 genera that I have, there are little tufts of hair in the final sternite. So I know that's a feature that I will use. So I start to get, detailed images of the things that I want people to be able to look at. And for me, I have to say, the photographs are pretty important. there's a lot of cases where a drawing is better. You can emphasize shapes and edges and so on. But what the user of a key is going to be looking at is what you see in a photo. So I like the idea of being able to get as close as you can to the experience of the person who's looking through a microscope. Next slide. So another step in the process for me is to stare at these images and think a little bit about whether there are shortcuts. And I'm pretty sure that specialists will be leery of this process on my part, because it's tempting to look at a picture and say, oh, I can tell that's a cantha because it has punctation on its head, and I don't see that on the other Ceratina here. But to be sure about that, I really need to look at a number of specimens. A lot of these things are variable. I think you have to be cautious about that. But it may be that if I'm looking at 8 species instead of 40, it may be that, in fact, there are some shortcuts. For sure with Ceratina arizonensis in the upper right, that's a no-brainer. They're black. They're very small and they're black. And they also have a single row of punctation just medial to the eye, and none of these others do. That's going to be distinctive. So arizonensis is going to be very easy. I'm going to try to think about whether the maculations on the clypeus are helpful, because anybody can look at that and figure it out. They're also pretty variable, but maybe they will be useful for the 8 species that I have. So next slide. The next step in this process will be to build the key, right? I've looked at all the features. I've thought about the dichotomous key. And there's an interesting thing that I'm realizing right now, which is that consider this key for big cats. My first couplet is it tawny or does it have spots or stripes? And then for each of those categories, I have another couplet. If they're tawny, do they roar? Then they're a lion. If they don't, then they're a puma. And ditto for spots or stripes. There's a way that I discriminate that, have to make that choice. So if I work through this dichotomous key, I don't learn whether leopards or tigers roar, right? Because that consideration, that feature, has not been something I had to think about since I'd already made my discrimination based on them having spots or stripes. So it may be pretty complicated to go back and look at all of my Ceratina and try to score them for all of the characters that matter. So that's something I'm facing. So next slide. This, if I was on my own computer, I would be glad to show you a little bit about how it all works, but I won't do that. So we'll go to the next slide. And then, and Claire, I have to say this technical glitch has been advantageous in that I haven't gotten into the weeds by looking at these things on my own computer. So I've been fairly efficient, I think. So really, this is where I just want to know what other people think about the niche for this. So as I've said, for me, there's a place where making a key interactive instead of dichotomous is going to be immediately useful to certain kinds of users. I think it's a good niche for limited region, my example here being the genera for the Willamette Valley, or for limited taxa for Ceratina because it's not super diverse. And then within project tracking, as the example I used was for Andrina or Megachile, so I can communicate about what's going on within our project. I wonder who would be a target audience for creating any of these sorts of tools, who would build them. And then I want to make sure that I consider the concerns. So I made up this new aphorism. So you've all heard the perfect should not be the enemy of the good, but it's important too that the good should be a mortal enemy of the mediocre. That is to say, I want to try to be practical and simple, but taken to an extreme, as you all know, B taxa are highly diverse, and you can't make a conclusion, for example, within genus about whether something has, is brown or black as opposed to metallic. You better know that for Lasioglossum, some of them are metallic, even though most of the ones people see most of the time are not. So there's that trade-off. Another concern that I have is stepping on toes. So for example, the genera of the Willamette Valley, there is already a dichotomous key that was created by August Jackson, and it's really good. And August, I'm sure, put very significant amount of time into it, and he's a legit taxonomy expert, unlike me. So I would want to be careful that I don't make a key that he would see as either encroaching on his turf, which, hopefully not, but he might also say, wait a minute, who are you? You didn't have my reference material. You've gone wrong on some things. This is too simplified, et cetera. So I have to be careful about that. And to be careful about that, I would want to vet whatever I produce. I'd want to see what August thinks about the key that I've made. It would be great if I could test unknowns and see if they come out the way that my key thinks they should come out. And then I want to know how usable it is. So, if my target audience is a citizen scientist, can they interpret what I've thrown at them in a way that's repeatable? So that's my whole thing, and I would be, delighted for any discussion or feedback or ideas.
Huzzah! And I think I in the email that I sent to everyone today, there were a bunch of the links that he's mentioning. So yay. And I just wanted to put in my 2 cents that I really love that you use Serotina as your example because they're my favorite, and would love to know a little bit more about your photography because that's amazing. And then I'll open it up to either Sam or the rest of the class to ask questions. But I'm using my authority to go first.
Great. And Claire, you're top on my list to vet my Serotina key then.
Well, I don't know the West yet, but please, I would love to learn. I will become that person.
Well, right. And then of course I have to say that I mean, I'm being both humble about this stuff, and not assuming that it's, the right solution to everything else. But if I did make a Serotina key that was useful for the Western species, there's no reason why that wouldn't be a template for you if you wanted to do Eastern species. Some of the pictures would be still useful. There's some overlap. So I think there's a way to make this, to make initial efforts that take a lot of time easier in another iteration.
Absolutely. There are ways where this is perhaps more usable and shareable than creating what we've been doing of these quick guide PDFs, which it can be a both and of using these and then also having PDFs that basically walk you through similar non-dichotomous choices.
Yeah, for sure.
Gonna open it up to other people, and we can just have a side conversation about your photographs.
Yeah, just a few comments from me. So I think the more guides there are, the merrier. I often get stuck, particularly on tricky groups, and I will look at someone else's guide or, pull up Mitchell or dichotomous keys just to try and see if I, using a different pattern of keys, come up with some kind of, at least a consensus as to where I'm going. A problem with guide development, though, is something that is that the person who develops the guide, you are a good example, de facto becomes an expert for that guide. And you have looked at their specimens a whole lot, and you've written a whole bunch of things, and it makes total sense to you. You have mind pictures of the characters, and you're writing things, and your mind goes immediately to the answer. And what we found is that that is like everyone doesn't have your same mind. And so experts sometimes can become the worst possible person to develop a key because it's like, what do you mean? It works great for me, but they already know the answers. So the best way to follow up is a couple things. One would be to take your advanced novice who wants to use microscopes and things like that, give them a set of serotina, in this case, where the name is disguised, and say, and best is if you're right there and like walk it through and talk out loud, like what you're doing, like, oh, I'm looking at this and, oh, I see it's got the, the pits next to the eye. So I'm going to click on that. And you can right away see where things kind of fall apart, right? Because the name that it should be disappears. So you get this kind of feedback, and then you can ask, well, why? What was it? Why did you choose that versus this? And a lot of times it's just clarity in the explanation. So a lot of keys tend to try and trim down the wordiness, but a lot of times what we end up doing is adding more words because people don't know the shortcuts and the little, what do you mean by blah, blah, blah? Well, you need to kind of explain that right in the key sometimes. The other, in terms of your ultimate outcomes, so, no key is perfect for everybody. So, and nobody walks into the taxonomy, like I need to ID things world and uses a key and gets everything right. So, it's some combination of how well the key is written for them and their abilities and, their mindset. So the way we look at things a lot is like, hey, try to identify your stuff. You're like an apprentice, and then bring it in and we'll double check it. And so you, me, and you and other people who are in the business of checking other people's work, that's cut down on a lot of the headaches, which is moving things around or having a complete hodgepodge collection. It's like, yes, yes, yes. Oh, that one's wrong. And we'll move that. But I have fewer things to move around. So I think like LINQ, for example, you can develop a bunch of keys. It doesn't mean that people are going to use them well. And there may be mistakes, or not even mistakes, but things that could be done better that get people to the answer better, that you will incorporate over time like we do. But ultimately, LINQ and others who are going to do your last check are going to be really thankful for anything that ups the average ID score. So yes to everything you've said, and I'd love to talk about getting your pictures, and then we'll host them on Discover Life too, if that's okay. And obviously,
of course, all
of our stuff is available, including pictures and things like that, is available to you if you want them, as is the guide characters and states, and I can show you, or anyone actually, how to get in there and grab all that stuff. And then the final thing is that it kind of looks like we run Discover Life guides, but there are many different kinds of guides in there. It's an open access thing. You can just get a blank guide, and it can be the serotina of Willamette Valley is totally fine, and do your own guide. You're not in competition with us. It'd just be one additional one, and you can use our platform, but it's got its own set of kludginess and things like that too, just like the ones you mentioned.
Right. So Sam, I'm listening very carefully to everything you say, because I'm a complete neophyte in this world, both in taxonomy and in creating, these sorts of resources. So I'm really appreciative of anybody else's ideas about how this can go. I'd be perfectly happy if I throw this idea out and somebody else says, please help me understand how those CSVs in the software work. And I don't have to be involved in it necessarily, but I'm pretty motivated to. I mean, this is great. And I think, tagging onto something you said there, you totally need novices and experts both. And my example of what you were saying initially about how you have your own mind picture, sort of a peeve of mine is arched basal veins. I'm going to tell you that serotina has arched basal veins, as does andrina in certain cases. And that anybody looking at a basal vein will call those arched, even though we all know, because we're on the inside, that no, that's a halicted character, right? So, yeah, yeah, you really have to figure out sort of, I mean, it's all about understanding your audience. And that's another piece of trying to figure out how to use these things is who is your audience? And I'm not sure. I've told you who my audience is. It's the people I work with, and maybe citizen scientists, because I spend a lot of time on iNaturalist, and it's frustrating to me because um, the ID process is clunky. A lot of times, you know, you get IDs that aren't warranted. So I always love it when John Asher comes along and looks at something I've ID'd to species and he says, no, genus, you know. But the images on iNaturalist are very rarely adequate to get to species.
Right.
You have to sort of guess, or you have so much experience, you say, oh yeah, you know, that's Rubiflorus because it just is. John Asher can do that. Sam, you can do that. I can't do that. So anyway, I'm thinking about citizen science and what we can do to get the person who posts and says, I think this is Lasioglassum mellipes, help them maybe know that it's mellipes. And I think it's both useful to make the database better, so we have finer grain information about what's out there, but also it's about empowering the citizen scientist. I'm way more interested in looking at Lasioglassum if I feel like I can identify some of them, and then I collect one and I say, gosh darn it, this is looks like mellipes, but it ain't because of X, Y, and Z.
Yeah, yeah. There's no one path here. So this is great, and you'll probably get some emails from people who want your help in helping set up their guide or whatever. And so yeah, really, the more the merrier.
Yeah, no,
And
I really appreciate that. So I'm not worried about stepping on your toes, Stan. Not too much.
I have to say that getting to the West Coast is last on our list because that's not our skill set. Our skill set is east of the Mississippi, and even there we have tremendous ID problems with certain groups. So yeah, don't wait for us. We're also happy if anyone wants to, and we're working with Alaska and Yukon now. And then Karen Wright just gave me all her Melisodes tables to expand any of the guides to different areas or get corrections and things like that. But we ourselves are not going to, we're sludging through the Midwest right now. But if someone else wants to do something or work with us on any of the guides, and it includes just a single state. So like Alaska, they only have 100 species, period. So that's great. So it makes it pretty tractable. And you can put in very conservative scorings just from the literature on a lot of these species. It's often clear from the literature that what the states of characters would be, even if you don't have a specimen. And then other ones, it's like, no, it's not even mentioned in the freaking description. So you can't score it for that, but that's okay. All these guides allow, well, I should say, all the matrix keys, like the ones you just talked about in Discover Life, you don't have to score completely each species for every single character because sometimes you lack material or, you just don't want to because it's not really appropriate for that species or whatever.
Yeah,
So there's a
absolutely.
lot of flexibility, and you don't have to wait for the perfect collection. You can catch up. Like we often go like, oh my God, here's Tracusa writing eye. We've never seen one, but now let's use it to double check the scorings. And like the arched veins and other things like that. Yeah, that's a classic character that is easy to misinterpret. And when you're dealing with matrix keys, you can what we call overscore. So
Sure. Yeah.
Nomia is an example of a halictid that really does not have a super arched vein. So we score it for yes and no on that one and so forth. So,
Right.
because elsewhere is a really good character. Like Nomia, come on, anyone can do that. And when you get to the pearlescent rims, except for that one species that doesn't have pearlescent rims. But I mean,
Yeah.
I want to open it up to anybody in the chat who wants to unmute and put in their 2 cents.
I suggested that you guys show David the differences menu. He may not realize he could pick out his 8 Serotina and look at those characters in Discover Life.
Oh yeah, Sam can do that.
Yeah, my thinking is that we're going to go through a couple different, like, who's up next week is the Wyoming guide, right?
Lucia's up next week, yeah.
Yeah. So, and then maybe after that, we'll go through Discover Life in the more technical things, like what you can use and what you can do and how things are scored, and some of the the nitty gritty things. But yeah, there is a differences section in Discover Life, often overlooked, that can contrast 2, 3, 4, doesn't matter, species, and it'll only show the characters that differ among them.
Yeah, I need to know about this, obviously. It doesn't jump right out as a feature.
No, it's in the menu. You have to click on the menu and look at differences and click on them. So it's worth repeating because, it's not a process that most people know much about.
Yeah. One other thing I wanted to mention is that an advantage of the keys that I'm working with, and I'm not sure how true this is for Discover Life, if someone tells me this afternoon that I'm wrong about something, I'll fix it tonight, and it will be live with the change.
Yeah. Yeah, Discover Life is similar. So I can go in, and we're constantly tweaking. People don't really realize that we're constantly messing the guides. And it goes up on what we call a developmental server the day so that I can work on it. And then it's pushed over to the public side at night. So the next day, whatever the changes were are live.
Yeah, cool.
So we get this question every couple weeks. So I think it's about time I made a PDF how-to with screenshots and whatnot. But this is just a quick, like, if you're in Discover Life, you go to your species, you click menu, or you go to your genus, you click menu up here on the top, you click one of these bubbles, and then you can use Control F or go through here, and it'll give you some greater control over exactly what characters you're looking for, rather than having to click through that typical guide every single time.
Yeah, because the typical guide, when you click through it, sometimes it doesn't let you see. You've already made some choices, and now other choices are not visible anymore. But I want to see those. Yeah, cool.
Yeah.
So the has character, the has function is good. The difference is you would click the radio button differences, and then click the selection box, the square box to the left of the species, and then click submit, and then it will show you a table of differences. So the has allows you to drill into, like, well, who has these characters? Because in a guide, you have things where some species are scored and some species are not scored. So has allows you to go in to find examples of things that have a character, and you can use only to find for a large character set that this species is scored as only having that one character. So again, it should be a good exemplar of that character.
Wait, before you go on, Sam, you're saying that only Serotina cockerelli has that one row of minute pits?
Yeah. So,
but so does Arizonensis, doesn't it?
Yeah, but this guide, I don't know which one and that was an interesting example you used, because we really have the names in, but for most, it's not usable for most of the West. So
gotcha.
We in our regions where we're actively updating species, we don't include Arizonensis, and you're right, it's in the same subgenus as what's its name, cockerelli, and would come out that same way on a lot of these kinds of characters.
Yeah. And so that's an example of something that we haven't scored for either. It doesn't show up with either one. It doesn't show up with more than one row of pits or one row of pits.
It's not.
Mhm.
I think Arizonensis is
Not horizontal.
just a name sitting in there, and at the top of the guide, it you know, you would almost all circumstances you're going to click on the. Sometimes it says Eastern only, and sometimes it says, I mean, as a like you can click for Eastern only and eliminate the rest of them, and other times you click on the state, and you know something's going to show up. See, species. If you look on that one, species under development, primarily Western. Well, it is way underdeveloped. Like no one bothered to score most of those, maybe all. I think someone started. I think we had another person interested. The thing is you're an exception. Like we have made this offer many times, and there's a certain person. You are an example of someone who likes all that little nitty gritty stuff. You know, taxonomists that get into it. Otherwise, it drives other people insane. I think, and people don't want to play that game, which is sad. But we're here to make things usable for the end user, and to go to that next level of actually dealing with the guide creation. Your mind has to be working on lots and lots of if thens and what about that, and yeah, you have to spend some time to wrap your mind around it, and a lot of people don't have that time or that desire.
So I want to mention one other thing, which is what I do is some field work on the projects that I mentioned, but that's not all my time. The rest of my time is I'm retired, so I do what I want, and what I want to do is look at bees under microscopes and spend a fair amount of time running around looking at actual bees in the field. So what I'm looking for, this key thing, I need collaborators to do anything really very useful, because I don't have the specimen collection and I don't have the expertise, but I have the motivation and a sense of how to use the tools and a reasonable ability to make good diagnostic photographs. So I'm sort of sitting out here waiting for anybody to say, okay, help me with X. And I'm open to that. The next thing after I do Serratina is I want to do Amphidioines, because there's someone in the Oregon Bee Atlas that's working on that. And she thinks I'm going to want to photograph genitalia. Oh my God, they're small.
Great.
So we'll see.
Also,
but I'm. Go ahead.
I was going to say, this is super useful if you're in a country that has, essentially no guides to bees or anything. So you can really start with your collection and create your own system. And then maybe later you get names on them. And like you have morpho species, same is going to be true in big chunks of the world.
Okay, well, we are at 2:06. Does anybody need a dash? Are there any more things from the participants who want to unmute? Okie dokie. Well, we're starting to get the things in the chat. David, I think that you and Sam and our guest Nancy will have an email thread about taking pictures. But other than that, I'm gonna wrap up the recording, and thank you very much.
Yes, thank you.
You're quite welcome. Very much.
And I put your email in the chat, so that you can also hopefully be approached by volunteers.
Yeah, glad to, of course.
Alrighty.
All right. Thank